This video will focus on how the molecular mechanisms of leptin, a key player involved in the biochemical pathways in obesity, lead to responses in the brain. Obesity is a condition that is associated with having an excess amount of body fat. Obesity has become an increasingly prevalent disease, with the obesity rates for adults shifting from 15% 3 decades ago to more than 35% this past year. Obesity can lead to many detrimental health effects such as heart disease, stroke, type 2 diabetes, and certain types of cancer. These are some of the leading causes of preventable death. From a scientific perspective, obesity is created by an imbalance between energy consumption and energy storage. Namely, the body stores too much. What controls how much we store, when we store, where we store, how we store, essentially everything, is the brain. More specifically, the hypothalamus is responsible for the regulation of energy balance, the brainstem for the actual metabolism of food, and the limbic system for pleasure and reward. These three tissues and their neurons each regulate complex molecular signaling networks. Let's focus on the hypothalamus, where the integration of various signaling networks in response to energy intake or energy expenditure occurs. Let's dive even further down to a special and crucial molecule, leptin. Leptin is a molecule or hormone found in the adipocytes or fat tissue that communicates to the hypothalamus to inhibit feeding and fat synthesis. The function of leptin and the obese gene has been studied in knockout mice that no longer have the gene. One of these mice was administered leptin and maintained a normal body weight, while the other was not administered leptin and was overweight. From this, it was concluded that leptin played a major role in weight regulation and metabolic homeostasis. Leptin acts on two types of neurons found in the arcuate nucleus through two different pathways. Leptin inhibits the appetite-stimulating neurons through the JAK-STAT pathway, which inhibits the release of hormones NPY and AGRP responsible for food intake. On the other hand, leptin stimulates the appetite-suppressing neurons through the PI3K pathway, which stimulates the synthesis of POMC, causing energy expenditure. Before we take a closer look at these pathways, let's examine our protagonist, leptin. Leptin is a fairly small 146 residue hormone with four main alpha helices that have an up-up-down-down folding pattern. This structure was initially difficult to crystallize as it had very few homologies in the protein database and had high tendencies towards aggregation. This structure shown, in fact, has a single mutation at residue 100 introduced to facilitate crystallization. The leptin receptor has 6 main domains, 4 of which are part of the immunoglobulin family and are shown in yellow and green. The other 2 domains shown in pink and purple are the leptin binding domains and, as the name suggests, interact with the leptin molecule. Once again, crystallization of the LBDs without the immunoglobulin-like domains has proven difficult as there are many flexible loops in the LBD structure. In fact, the study showed that one of these highly flexible loops is the key to the binding of leptin to the LBD domain. In the JAK-STAT pathway, our leptin molecule binds to the leptin receptor, causing it to dimerize, and activate a Janus kinase to phosphorylate the receptor at 3 specific tyrosine residues that then activate 3 separate pathways. Each of these residues become docking sites for different STAT proteins. These STATs then become phosphorylated by JAK and dimerize. In this form, they can move to the nucleus to modulate transcription factors for POMC and AGRP. STAT binding leads to opposite effects for these 2 genes. In POMC, STAT binding makes the DNA accessible and leads to enhanced expression. On the other hand, in AGRP, STAT binding makes the DNA less accessible and leads to reduced expression. Without leptin present, the appetite-stimulating signal is predominant, but with leptin, the appetite-suppressing signal predominates. How is leptin and its regulation affected in people with obesity? People with obesity show resistance to leptin, meaning that even with high levels of leptin, they are not able to maintain a normal body weight. One mechanism through which this may occur may be caused by a defect in the JAK-STAT pathway. The pathway is negatively regulated by a protein called SOX that are expressed when the pathway is turned on. They bind to one of the phosphorylated sites on the leptin receptor or directly on JAK and inhibit phosphorylation by JAK. In recent studies, it has been found that mice with diet-induced obesity have a defect in this pathway, although the exact effects on the SOX molecules have not yet been determined.